Chapter 12 picks up basically where Chapter 11 left off. So in the last chapter, what we were looking at was how we got from chemistry-related chemistry, methyl groups, carbon dioxide, really simple stuff, to biochemistry, amino acids, DNA, RNA, maybe even the first living cells. Where this chapter picks up that continuation is, so now what happens to those early cells? How do those change? What are the major big kind of leaps forward in evolutionary history? And we get to start by talking about slime mold. And this is actually a diatomaceous algae that you're looking at. The cell walls are actually made out of silica, so very similar to glass. Some slime molds can actually live singly as individual cells, and others can live as parts of multicellular groups. So, spoiler alert, this is one of the things that we look at is how did we get from single-celled organisms like amoebas and really simple protozoa and bacteria and stuff to the complex multicellular us's that are walking around. So by looking at organisms that have life stages of both single-celled and multi-celled, we hope that we can maybe start to get a handle on how those transitions may have occurred. So slime has a kind of a different life cycle than humans do. And it starts off basically, the spores actually are released into the air, onto the animals, plants, whatever, leaves, etc. They find a new spot in the ground and they spend some time as single cells. During this stage, they're called amoebas, not amoeba like the ones you've thought of before. They scavenge through the soil and they eat bacteria during this part of their growth phase. Eventually, as they start running low on bacteria, it triggers a biochemical change in the cell, and the cells actually start to come together and they cluster together and they aggregate and they form a multicellular slug. Now, this thing is actually better at moving through the environment, we'll look at that later, and it's able to scavenge farther, faster, and better than the individual cells were. When it comes time for reproduction, after, again, you start getting kind of low on the number of available bacteria to eat, these multicellular slugs start to form a fruiting body. So they grow this kind of long stalk with a little ball on the top of it, and that little ball produces the spores that float off into the world. And that starts your whole life cycle over again. What's interesting about this is that the fruiting body, the head part, is the only part that actually functionally reproduces out of this whole thing. The rest of the stalk and the base actually shrivel up and die. So from an evolutionary perspective, if survival and reproduction is the name of the game, that part, the part that dies, isn't really getting a whole lot of benefit, or at least it doesn't look like it. So that's something that we have to try to tease apart is, why is this advantageous? Why is this actually favored? Why do the algae and the slime molds do this? And that's part of where we're going in this chapter. We've also noticed that through evolutionary history, both sizes of organisms and their complexity increase over time. So this is going back all the way basically back to the beginnings of life on Earth. It stayed pretty simple and pretty primitive. And then all of a sudden it started to get physically bigger. And it started, this is the physical size thing, and we started to see the complexity increase. So the number of different types of cells that you would actually see in these organisms also increased over time. We even see this if you go back and look at the dinosaurs. The first dinosaurs were actually fairly small little critters, and they got bigger and bigger and bigger and bigger and bigger until basically the KT extinction event. We also see the same trend in terms of complexity. The longer something is around, the more complex those organisms tend to become. And we are really good examples of some pretty complex organisms. So in this chapter, what we're going to be looking for are, what are those major transitions? What are some of these big, huge, like life-changing types of things? How do we explain those? Why was just the development of eukaryotic cell types one of these? Multicellularity is another one that we'll be looking at. Individuality and solitary to group living. So this is already basically most of the list of what we're going to be covering in this particular chapter. So we start off with how did we get from bacteria, prokaryotic cells to eukaryotic cells, including some new ideas that I get to tell you about that haven't previously been included. When and how did sexual reproduction start? What gives with the sex thing? Why is it beneficial? We'll deal with more of that in the chapter that's devoted to sexual reproduction, but we get to start looking at it here. What about multicellularity? When and how did that get started? And what about individuality? And why is that after multicellularity? I mean, a single cell is an individual, right? So why is individuality different than multicellularity? And why do organisms, some organisms anyway, live in groups? Solitary to group living transitions. So this is kind of where we're going. Are there other big changes that have happened? Sure, but these are some of the most fundamental and some of, in fact, the hardest to explain. So starting with how we got from prokaryotes, our simple living cells that we sort of had a tenuous grasp on at the end of the last chapter, how do we then get to eukaryotic cells? We start seeing them show up in the fossil record around 1.8 billion years ago. And we started seeing the first life forms about 3.5 billion years ago, based on things that we've looked at before. So it took, that's about 1.5 billion years of prokaryotes only before we started to see eukaryotic type fossil remains. These are cells that have mitochondria and or chloroplasts. The major criteria is membrane-bound organelles. And that's important. One of those is the nucleus, but it also applies to other membrane-bound organelles such as the Golgi complex, the ER, the mitochondria, the chloroplasts, the lysosomes, peroxisomes, fill-in-the-blankosomes. I made that one up. All right, so when you learned about these different cell types, one of the things that you probably remember thinking is these are kind of complicated. And yeah, they really are. When you take cell biology someday, you'll get a much better appreciation of how complicated, because we have to look at all of those cell parts. But trying to coordinate all of these activities, transcription, translation, glycolysis, the Krebs cycle, all of these things going on in the cell, protein synthesis, protein degradation, bringing in glucose, coordination among all of these things, including all of the organelles, gets really complicated. So what favors that additional level of complexity? How did this start? One of the things that we're actually a little bit less sure of than we were a decade ago is whether we should have 3 domains of life, which is what most of you have been taught for a long time now, or 2 domains of life. Now, this is actually kind of a weird question, and it's one that made me really excited when I was looking through the second edition of the textbook, because that's where this started to show up. Currently, we have 3 domains of life: the archaea, the eukarya, and the bacteria. So this blue box in here, those are basically the archaea. And that's what we've all been taught, 3 domains of life, right? It hasn't always been that way. Even in my lifetime, this has changed. When I started college, which was a while ago now, we were actually still debating whether or not this should be included in textbooks. The jury was still out on this, so to speak. The original research was done in the 1970s by a group led by Carl Woese. We talk about him some more in cell biology, and you actually get to look at some of the data from this. But he sequenced ribosomal DNA, and he compared that across a lot of different kinds of organisms. And what he found was that the eukarya kind of clustered together. Their genetics for this ribosomal DNA were similar. The archaea clustered together. They were more similar to each other than they were to anything else. And the bacteria formed a 3rd cluster. And that's where our 3 domains of life idea came from. Except we've found some more things that were kind of interesting about this. There is another hypothesis, and it's been around since at least the 1980s. So it's a little bit more recent, but not a whole lot more. I found additional articles from 2008 and 2013 that also that looked at this, and it's the eocyte hypothesis, the second figure in B. Now, this one actually suggests that we have only 2 domains. We have the bacteria, there's the bacteria, and we have basically the archaea, and that eukaryotes are actually a subgroup of the archaea. That's weird, because that would mean that we evolved from archaea. So we know where the data for this figure A came from. That was the ribosomal DNA analysis done by Woese. What evidence supports the eocyte hypothesis, this other idea? And that is something that is significantly less discussed in most evolution classes. In fact, this is the first time I've ever even really had a chance to talk about it. So I'm kind of excited. To be honest, I totally nerded out when I was reading about this, and my boyfriend kind of made me go away for a while. Yeah, he didn't want to hear about it. But you guys do, right? Because this is cool. This is like modern and up-to-date stuff. So here's actually what we know about these different trees of life. I have to justify what evidence supports the eocyte hypothesis as opposed to the three-domain hypothesis. And this is something that's still being debated, and one of the articles that I read said it probably won't be resolved in our lifetimes, if ever. And part of that is just because we don't have a time machine. We'll never be able to go back and really observe this. So our early studies on RNA and enzymes and ribosomes all showed that without a doubt, prokaryotic and eukaryotic cells are related. It doesn't matter if you're talking about bacteria or archaea or eukaryotes, we all basically do glycolysis about the same way. We're related. Our DNA is similar. But our informational genes, the ones that code for our proteins, that tell our skin how to make collagen, that tell us how to grow hair and make brain cells, those gene sequences are actually more closely related to the ones that we see in archaea today. And this is kind of surprising. In cell biology, one of the things that we actually look at is the ribosome structure. And it turns out that the eukaryotic ribosome looks almost exactly like the archaeal ribosome. So this is stuff that is kind of in support of the eocyte hypothesis from the previous slide. But here's what's also weird. Our operational genes, the ones that tell us how to make a cell membrane and the ones that tell us how to make amino acids and some of these really fundamental stuff, they are actually more closely related to eubacteria. What does that make us? What are we, some sort of weird hybrid? The answer to that is actually maybe. There are some camps, some groups of researchers who actually think that the nucleus itself might actually be an endosymbiont, a lot like the mitochondria and chloroplasts. So that's what this last part down here is actually trying to get at. For one thing, our cell's nucleus actually has a double layer of membrane around it, like the mitochondria and the chloroplasts. For another, our genetics appear to be a fusion or a hybrid. We've got kind of a mix of both bacterial genes and archaeal genes. So maybe we are actually kind of a strange hybrid of both. There is one more notable difference that I came across in some of those readings from the 2013 article in particular. The way we actually put our cell membranes together is actually more similar to bacteria, to the eubacteria, the ones that we generally just call bacteria, and it's actually not the same as the archaeal cell membrane structure. There's even slight differences in the chemical nature there. So that was weird, but kind of cool. So I want you guys to be aware that these articles just came out in the last like 3 years from the time I'm doing this recording. And this is a change over what I've been taught. I had to go back, look stuff up, and try to figure out, does this fit in with what I already know about the genetics of what's going on? Does this fit with anything that I've learned or been taught? Does it change everything? And this is one of my favorite parts about science, is the ability for us to actually look at evidence and go, oh wait, that makes sense. And that makes sense. Okay, so maybe we've got two possible explanations. Maybe we've got a fusion of possible explanations. Again, no time machine, so we'll never really know. We do know the following things about endosymbiosis, though, and we have been able to do a lot of research on this. So if you take a really primitive archaebacteria, archaea, over time, somewhere back in evolutionary history, somewhere around 1.8-ish billion years ago, we think that it engulfed an ancient proteobacterium, and this actually created the first endosymbiont and our first eukaryotic cells with mitochondria. So this is where our mitochondria came from. Later on, that now eukaryotic cell engulfed something else, a cyanobacterium, and that gave us all the plants. What that actually means is that plant cells are a more recent development than, strictly speaking, other eukaryotic systems. They are a little bit more sophisticated. They have an organelle that we don't have. So some of the evidence that supports this idea is the idea that chloroplasts and mitochondria both have this double membrane layer, and this is actually normal. So when you start off with a cell, as another cell tries to enter, or a virus or a piece of food or anything, what happens is your cell membrane actually kind of bends in and kind of covers and engulfs that particle, whether it's food or whether it's a bacteria or whatever. So you temporarily end up with this membrane enclosed little bubble. And what we kind of think happened is that instead of digesting whatever was in that little bubble, our proteobacterium or our cyanobacterium, basically the cell kind of went, oh wait, I forgot to do that. Well, hey, that thing's over there making energy for me. Maybe I should keep it, kind of like a pet. That would be awesome. And it stayed that way. It was very, very efficient. It was very favored by natural selection. It ended up being a really good symbiotic relationship. In addition to that, both chloroplasts and mitochondria have their own unique DNA. We can isolate that and we can sequence that. It's been in crime shows for years now talking about mitochondrial DNA. They don't talk about chloroplast DNA so much, but it also exists. And when we sequence those genes from chloroplasts, we find out that they look like cyanobacterial genes. They map very, very nicely. They correspond very well to the cyanobacteria that exist today. Similarly, the mitochondrial DNA looks like proteobacteria today. So that's why we think that these were probably originally free-living organisms that kind of got not totally eaten. The last part of this puzzle that we're going to deal with for this particular chapter is that both mitochondria and chloroplasts can also replicate independently of the host cell. So one of the things that happens when you work out, let's say you have to run away, the zombies are chasing you, must run. The first rule of the zombie apocalypse is cardio. You don't have to be the fastest person, you just have to be not the slowest. Okay, when you're running, when you're practicing your running, when you're training to run, I like practicing better. One of the things that happens is when you first start out, you're not going to be able to run very fast, very far, or very long before you're like keeling over going, air. That would be me most of the time. I'm not good at running. As you do this more and more often, your body adjusts to it. And part of that adjustment is that the mitochondria in your muscle cells actually replicate. As you put more energy and oxygen demand on your tissues, your mitochondria in your cells will be stimulated to divide and make more copies. This allows them to process more incoming glucose, provide more ATP energy, so you can keep running and not hit that wall of exhaustion quite as early. If you stop exercising for a period of time after that, those mitochondria will naturally sort of dwindle over time because they create a lot of toxic sort of byproducts, cellular toxins, not acai berry cleanse toxins. We don't even go into that because that's pseudoscience. Okay, so the same kind of thing can actually happen with chloroplasts also. The numbers of those actually vary, and in particular, they vary with the seasons. As I'm recording this, it's in the fall semester, the leaves are changing outside. Part of that is because the chloroplasts are starting to die off, the chlorophyll gets reabsorbed and destroyed, and the other cell pigments actually come out and you can see them better. So that's why the leaves turn reds and oranges and yellows. Cool, huh? So I mentioned that some think that our nucleus is actually an endosymbiont as well, or a fusion in some way. And part of that is because some of our nuclear DNA looks like archaeal stuff, and some of our organelle-based DNA actually looks like eubacteria. But we do actually get some bleed through, and this is where things get a little bit strange again too. In the last chapter, we talked about the idea of horizontal gene transfer, and that was the ability for organisms, especially bacteria, to transfer genetics directly from like one cell to the other cell without going through reproduction. That's what the conjugation pilus was for. It just forms a bridge where you can go, okay, you've got drug resistance genes. So what researchers did in this experiment that's being illustrated here is they actually took a gene for antibiotic resistance. It's the neo-STLS2 gene. And they put it in the chloroplast. They did that on purpose. It doesn't work in the chloroplast, so this accomplished nothing. And if you treat the plants with high levels of kanamycin or another antibiotic, you can even kill the plant cells with that. So that cell is dead. But if that gene successfully jumps and transfers into the nucleus, then it can become active, and the whole plant cell becomes resistant to kanamycin treatment. So all you have to do is throw a bunch of kanamycin at this, and whatever survives has those resistance genes that have successfully jumped from the chloroplast into the nucleus. And that happened about 1 out of every 16,000 cells. That's actually kind of a lot. So we have 1 trillion human cells or more. That's 1,000 million. Okay, that's a lot. And 1 out of every 16,000, that's actually a surprising number of cells where this would be happening in us too. Estimates are that everybody, you, me, everybody, has somewhere between 300 to 600 genes that have actually leaked from our mitochondria into our nucleus. And we function just fine. So there is some sort of degree of horizontal gene transfer or lateral gene transfer going on in there. So you may find yourself going, that's great. This was all like 2 billion years ago. Why do I care? Actually, there are modern implications for this, including some medical and drug treatment related implications. So this is an example of how we can actually use evolutionary history to tackle a modern medical problem. Malaria continues to affect a lot of the world where we don't have like lots and lots of anti-mosquito sprays, which we use widely here in Hampton Roads. So what if you don't have tons of pesticides, like in the tropical regions and in third world countries? Malaria is actually caused by an intracellular parasite. So this parasite infects you, it gets into your red blood cells. And this is actually kind of like what's shown in the diagram. So originally, here's what we think happened, because we noticed something weird about those malaria parasites. They have this organelle that isn't present in any other kind of organism. Not prokaryotes, not eukaryotes. They have their own extra little organelle. They're technically eukaryotic. They have a nucleus and mitochondria and stuff. But they also have this thing called the apicoplast. And here's what they think happened. So they think basically that a host cell, this big red cell here, engulfed a cyanobacterium. So this creates something kind of like an early plant cell. And then this whole thing got engulfed yet again by a bigger cell. So what we end up with is basically an organelle that has a quadruple membrane layer, 4 layers of membrane around it. And it's got its own genetics. And it's unique to this apicoplast family that includes malaria parasites. So can malaria totally do photosynthesis? No, it's not that lucky. Those genes seem to be non-functional at this point. Darn it. I think it would be so cool if we could do photosynthesis. I'd love to go out in the sun and be able to recharge. Oh, that would be great. So what does that mean for us medically? Well, currently how we treat malaria is with anti-malarial drugs. And these are designed to kill eukaryotic cells. Unfortunately, that means they have a lot of side effects because they also kill your human cells. So what if we could develop a drug, a treatment, that specifically targeted just this apicoplast, since we only really find it in malaria and closely related species of similar parasites? Here's what we know about it and about its genetics. This is kind of the structure basically of the malaria parasite itself. And this is the nucleus. You got some endoplasmic reticulum, and you've got the apicoplast being shown there as kind of the purple globby thing. We've actually isolated DNA from that and started working on sequencing it. Of those, we've been able to figure out genes that we kind of think we've got an idea of what they do, and that's less than a third. The other slightly over 2/3 of them, we have no clue. So that's not overly helpful. But of the ones that we do know, what do they do? Let's see, 25.9%, the single biggest block, those are involved in translation. The next biggest block, 15.9%, that's actually, let's see, protein turnover, protein import, post-translational modifications, miscellaneous. That's helpful. Why not just call that unknown? All right, next biggest block, 15.3%, making fatty acids and lipids. All right, I'm not sure that's a great target. 8.8%, DNA replication and repair. Okay. I mean, if you look at these, they're all fairly normal cell functions. Transcription, translation, making proteins and recycling them. Nucleotide modification for making DNA and RNA stuff. This isopentenyl diphosphate synthesis, maybe a good target, maybe not. These iron sulfur assemblies, maybe a good target, maybe not. So what we're hoping is that as we sequence more of this, we can find a gene that we can use to kill the malaria parasite without affecting us, because we wouldn't have that gene. This is still something that's being actively researched. The last I've read, they had developed some treatments, but most of them hadn't been overly successful. I don't think any had made it to human trials yet, which is kind of sad. But maybe. I mean, it never hurts to try and see what we can come up with. So as we move on to the next stage in this, we get to multicellularity. And when do organisms go from being single-celled to being multicelled? And we tend to think of this as being pretty absolute. You know, organisms either are single-celled or are multi-celled, and that's not exactly fair, because we already saw in the chapter beginning thing with the slime molds that there are some weird freaky things that can kind of do both. It's not common though. So as we talk about this, we have to start thinking about this issue of scale and economies of scale. And what that refers to is the idea that the bigger and more complex an organism is, the bigger and more complex problems it can tackle. So for this to work, for there to be a good movement toward multicellularity, for example, individuals have to give up the ability to reproduce independently, and they have to join together to form a larger grouping that shares reproduction. This is what we start to think of as being a multicellular thing. And we saw that with the slime mold. We see that with the ants. We see that with us. We are large multicellular things, and not all of our cells can create new human life. My skin doesn't just bud off and make a baby. So these aggregate multicellular individuals can tackle bigger problems, and these are problems related to scale, which is what you're looking at in this particular figure. So you've got one ant by itself that would not be able to drag off this nice big worm, but a bunch of ants working together can. So this is an issue of scale. Specialization, and when you're looking at social insects like ants, you've got the queen ant, you've got worker ants, you've got defender ants, so they become specialized. Even in our bodies, we've got specialization. We've got heart tissue and brain tissue and lung tissue. So this is what we refer to as economies of scale. And the whole idea behind this is, again, the bigger and more complex something is, the bigger and more complex problems it can address. This also offers us new and better ways to process, transmit, and store information. And I'm going to have to go back and relabel that. Economies of scale. So the bigger and more complex organisms become, the more likely they are to be able to devote some cells to things like memory and communication and hunting behaviors and foraging behaviors and all of those other types of things. So they can tackle bigger problems, and they can pass that knowledge on to other individuals or to their offspring, and that's where the transmit part comes in. So getting bigger and more complex should be favored by natural selection. And that fits well with what we've already kind of seen, which was that increase in both size and complexity. So for this to work, this is the general process that we kind of think things go through. The independent individuals first start banding together to form a new individual. And this is like those slime mold cells starting to come together and aggregate with each other. This new individual has to be more fit for natural selection to favor this. So it has to be better at survival and or reproduction. Otherwise, natural selection is going to weed it out. So we have to be able to explain how and why this happens. Why is this more beneficial to the individual than solitary living? We also have to be able to explain another thing. Why isn't there cheating? You guys have all observed this in group work, right? Where you've been assigned part of a group project and somebody in your group doesn't do their part of the work, and on the last day you find yourself having to do it for them because your grade's the one that's on the line. Why doesn't that happen in nature? Actually, it does, a surprising amount, but there has to be some sort of penalty to that. So what is to keep this cell basically from going with this other cluster, getting all the resources and all the benefits in the food, and then taking off with that, either stealing it or just using it itself for selfish reasons and going off and reproducing on its own? That does happen. But there seem to be some ways to sort of compensate for that and offset some of that. And some of that actually seems to occur with, I'm going to use a term here that's not great, biological programming. We're going to talk about this more in the next chapter, so we are going to kind of come back to this idea. But there are ways we can actually change how we use our existing genetics. So we don't have to change the ATGC sequence, but we can change how we use those genes. Like, which genes do we use? Do we use the genes for growing red hair, or do we use the genes for growing gray hair? That's kind of what I'm talking about. So when an individual joins a group, we often see genetic shifts. Which genes are active actually changes because they're part of a group. And it actually seems to turn off, in many cases, their ability to independently reproduce. That helps prevent some of the cheating issue. Like I said, let's kind of start at the beginning on this. We'll get into the how and the why of some of that in the next chapter. But how did this begin? So just to give a broad overview, what you're looking at is basically multicellular life forms and unicellular life forms and how often those occur. And this is among basically eukaryotes, all of the eukaryotic groups. So the red dots represent life forms that are purely multicellular. And let's see, we've got animals. Animals are multicellular. Yay, that's us. Wow, we're an itty bitty branch. Let's see, dictyostelid slime molds, plasmodial slime molds. Ooh, slime, that's exciting. Land plants, trees and grasses and flowers, and okay. And some of the red algae. That's 5. Ah, that's a bad 5. Okay, let me rewrite that. That's 5. Let's see, the yellow ones, those are groups that have both unicellular and multicellular forms. So we have the choanoflagellates, the fungi, the protostelid slime molds, chlorophyte algae, charophyte algae, and another group of red algae. We've got some oomycetes, brown algae. That's, I think that's pretty much everything. 1, 2, 3, 4, 5, 6, 7, 8. Not a lot of multicellular life. The green ones, mostly unicellular with a few rare multicellular forms, diatoms, ciliates. Diatoms and ciliates. 15 groups out of all of this. The all multicellular, the blue unicellular onlys, there were 31. And for what it's worth, I actually missed a yellow group over here, the acracid slime mold, so that's actually 9. So, okay, 16 total that had some multicellularity and 31 that were purely unicellular. It's a ratio of 2 to 1. We are a distinct minority. Okay. So what you're actually looking at is a form of multicellularity that occurs in yeast. And this is kind of cool. I've never actually seen this. So now I got to go home and actually get some bread yeast and I actually make my own yogurt so I can compare the two different versions of yeast. And I want to see if this actually happens. So at the start of the experiment, they actually took yeast that were purely unicellular and they started watching how they started to cluster together into these snowflake-like assemblages. And these are like the ones shown. And this is over the course of, that's 2 hours, 3 hours, 4 hours, 5 hours. So these clusters get bigger and bigger and bigger. Okay. When they get big enough, they actually seem to start dividing, which is what we see probably around the 5-hour time point. So yeast is generally considered to be unicellular. I'd never actually seen or heard of it behaving in a multicellular manner before. So like I said, I kind of want to do this at home. I know, I'm a total geek. I'm going to have to take a microscope home and just find out for myself. Back to the slime issue though, we can actually observe this more easily in slime. And part of what causes the slime to start aggregating. I mentioned before that one of the things that triggers that is when it runs out of bacteria in its field, so it has nothing left to eat. As the food levels go down, the levels of this particular compound in the cells, cyclic AMP, that starts to go up, so they kind of go in reverse of each other. Cyclic AMP is kind of like ATP that has lost 2 phosphate groups, except it goes back and seals into a circle. You learn about it in cell biology and organic chemistry and stuff, but for now, basically it forms a signaling molecule. It's not a normal breakdown product of ATP. So this is how the cells actually sense that they're running low on food. Your cells do the same thing. So as these cells produce more cyclic AMP, it also triggers them to start making sticky surface proteins that allow these cells to adhere to each other. Think Velcro. And once this has happened, they can actually migrate toward different stimuli, such as light or food. So what we're looking at in particular in these bottom sets of panels in A over here, what you've got is a worm, that thing in the middle that's bright green, that's a worm, and all these little individual dots, those are little individual cells that are of the slime mold. And you can see that they are much, much smaller than the worm itself is. So by themselves, as unicellular forms, they are not capable of tackling that worm and eating it as food. Now in B, this is actually reversed. This particular spot right here, that's your worm. This particular big thing, that is your aggregate slime. That is your multicellular slime arm, so to speak. Bye-bye, worm. It becomes lunch. So this is, again, an example of economies of scale. Because the slime have actually, the cells have aggregated together to form this multicellular slug, it can actually tackle prey that is significantly larger than the individual cells were. And therefore that affords it more eating opportunities. It can now use that instead of just having to rely on bacteria. Interestingly, some really bored scientists apparently decided to have slime races. Does this not sound absurd? So here's what they did. They took some dictyostelium slime mold in both single-celled form and slug form. So unicellular and multicellular versions. They stuck them in a petri dish with some dirt in the middle, and they put a light source at the other end, and then they had slime races to see which version got to the light first. This sounds like the most boring race I can imagine. What they found was that the multicellular slug was actually more efficient at streaming through the soil and reached the other side of the petri dish earlier than the individual cells could. So there was a clear advantage in terms of being able to move, in terms of being able to seek out new food sources or whatever the light source is kind of triggering. And these had a definite advantage over the single-celled versions. This helps explain, at least in some part, why they would band together. But my other question is, why are they responding to light? Okay. A lot of the dictyostelium slime molds and a lot of slime molds in general are capable of photosynthesis, so that actually makes some sense. They also put some bacteria, food source, over here by the light. So they should be following basically a chemical gradient that says food is in this direction and light is in this direction, so they crawled toward the light. That's kind of logical. Over the next 3 slides, what you're going to be seeing are some videos of skin cells, mouse skin cells, that actually move toward a light source. So I want to talk about those before you watch them so you know what to look for. But what you're going to see is kind of a squiggly thing. That's going to be your cell. I'm not very good at drawing them. And you're going to see a very small little dot, and that dot should actually be flashing. What this is an infrared light source. It's just like a little LED stuck in a board, and it flashes and it gives off a little pulse of infrared light. So what's interesting about this is that the mouse skin cells are actually going to migrate toward that particular light source. Now, how and why this happens is an interesting question, because it's not like the cells actually have a brain. They can't reason out, let's go check that thing out. It looks for all the world like they're curious, like they want to reach out and kind of say, hey, what's that thing over there? And they want to touch it. But they can't they can't think, they can't plan, because there's no brain in there. So this is all being driven by photochemistry, and it's kind of an odd scenario that we don't understand very well yet. So as you watch these videos, that's what I want you to pay attention to, is look at where the light source is, which will happen before the video starts to play. It should show you where everything's going to be, and then follow what happens to the cell. Now, each of these 3 videos is set up in kind of increasing difficulty for the cell to actually reach that light source. So the first one is pretty straightforward. In the second video, the cell's actually initially moving away from the light source, and it then has to kind of turn around and come back to the light source. And in the third video, there's actually kind of an obstacle between the cell and the light source. And they're not all laid out like this. This is just kind of my quick example of what to look for. So what you ought to be able to observe is the cells, even basic fibroblasts in your skin, actually do respond to light gradients. And this is something that we don't understand well. This has been published on very, very sparsely. It's been several years ago now when I was working in the dermatology practice that I remember reading an article about this. One of the biggest questions is, how does a skin cell, for example, detect light and dark? And it turned out that what they had found was that the skin cells actually express some of the same types of proteins that we use in our retinas. Not all of them. So it's not like we've got a bunch of bajillions of little eyes, but some of those light sensitive proteins still seem to be expressed at lower levels in our skin, but they're still there. So this may represent some of the physical basis behind how our skin cells respond to light. And this at least partially explains why the slime molds would be crawling towards the light. That and the bacteria and the photosynthesis aspects kind of make it much more self-evident. But it's interesting because of the levels of increasing complexity. So take a look at those and think about what's going on in those videos. Okay, so this is an up-close view of the fruiting body of one of those slime molds that we talked about back at the beginning of the chapter. What you can see is you've definitely got that stalk thing that's kind of growing up there. Let me change colors and try that again. There we go. Stalk thing growing up, fruiting body at the tip, and you'll notice that this connection right between here is really, really thin and really, really weak. It's actually that way to improve the dispersion of the spores that are going to come out of that headpiece. So as an animal walks by or a leaf brushes by, those spores can actually be transmitted for long distances. Now, that is hopefully going to be relatively important, because the reason that these things formed the multicellular slug in the first place was because they were running low on bacteria to eat. So by dispersing their spores over a larger area and farther away from their existing territory, they're hoping, well, in as much as slime can hope, but basically the function of this is to ensure that some of those spores land in more nutrient-rich areas. And hopefully they have a better chance of surviving and reproducing in those new areas with more bacteria to eat, because they've already depleted the area they're at. So this brings us to another question, and that's, when is a group not a group? I mean, I don't refer to myself as an aggregate of trillions of cells, at least not outside this class. I am me. You know, my cat is my cat. Your children or your grandparents, they are individuals. Ants are individuals. The mouse on my mouse pad, that's an individual. We don't refer to them as collections of cells. So where do we stop having just a collection of cells that are kind of stuck together and start having a functionally multicellular thing? Because some of that slime mold, it just initially, especially in the early stages, it was really just kind of an aggregate of identical cells. So Michaud's definition says that individuals are integrated and indivisible wholes that can reproduce and pass on their heritable variations. Let's break this down. Integrated. Am I integrated? Let's see, I have muscles, nerve cells, bone cells, stomach stuff. Yeah, I'm pretty well integrated. All of that works together as a nice system. Am I indivisible? Pretty much. If you cut me in half, I'm not going to survive that one. If I take off a piece of my skin, can I keep that alive in tissue culture? Yes. Now here's where things get a little strange. People ask me about this in my regular classes, so you guys are probably curious too. If I actually take a piece of skin from a person, like a large block of skin, I can keep that alive in culture for maybe about a week. And we used to do this in dermatology because we wanted to see whether we could use that for testing of like skin creams and stuff. And we would occasionally get donated skin from people who got tummy tucks. They don't want it back. The whole point of the procedure was to get rid of it. So they usually are quite willing to donate that for research purposes. A week is not a very long time to get to work with these cells, and then the whole tissue actually starts to degrade and degenerate and die off, and there's really nothing you can do about it. And I did that experiment like myself. I've done that in the lab. Now, if I were to actually take the skin cells out of all the extracellular matrix and stuff and put them in culture, I could keep them going much longer, which is a little ironic. And I might be able to get them to successfully divide and replicate somewhere between 20 to 50 times, depending on how old the person is who donates the cells. So the thing is, though, that those cells that I take, they're not capable of producing another human being out unless we use like cloning technology on them. You're not going to be able to isolate my skin cells and put them in a petri dish and expect it to make a baby. It doesn't work that way. So in that sense, yes, I am an indivisible whole. If you take away part of me, it dies without the rest of me being attached to it. That aggregate thing has to be able to reproduce and pass on its genetics, basically. Okay, we meet this definition pretty well. So what's interesting is that what we're looking at here is Volvox. This is a particular type of algae. It used to be really common in intro biology labs. And what's interesting about this is that some forms of Volvox, like the ones you see here in A, are single-celled organisms. This is actually panel A, and that one's actually panel D. There we go. So in A, we're looking at single-celled versions of Volvox. In B, what we're looking at is another version of Volvox, and this one is multicellular, sort of, but I don't know, it just looks like a cluster of cells to me, and it's about 8. You think that's multicellular? I'm not horribly convinced. Okay, that's actually not uncommon. This is a cluster of 8 to 32 undifferentiated cells. So all of these cells have no like tissue specialization or anything. In fact, we see something kind of similar in C. This is also a set of undifferentiated cells. It's just a little bit bigger, and this is yet another strain of Volvox. But we are starting to see that they are starting to enclose themselves in this little protective membrane sac-like thing, and that's what makes them a little different than what we saw in B. So this is getting closer to a multicellular entity. It's actually segregating itself from the rest of the everything else's with an extra layer of protection. In D, this is where we actually start to see real changes. It's not real obvious at first, but see these little tiny dots and those bigger dots? Those are different sizes of the same kinds of cells. And as it turns out, the bigger cells, these are reproductive cells. And the little tiny ones are somatic cells, body cells. So this is where we actually start to see a division of labor. This is part of that economy of scale. We've got tissue differentiation beginning in a really early way here, where there's a lot of reproductively capable cells, but some of them are strictly there for like physical support structure. That's kind of neat. And then if we look at E and F, we see even greater differentiation. So again, the larger cells, those are the reproductive cells. Those cells are the reproductive cells. So what we're seeing are differing ratios between single cells, multicellular, but basically no differentiation, to early and even late differentiation with lots of body cells and fewer reproductive cells. We've got this whole spectrum that's present in Volvox. And this actually tells us a lot about the stages that multicellularity might have gone through to evolve, if we can actually see all of these as individual steps in a process. And it's cool that we've got living examples of all of these. So that was pretty short and sweet, but when is a group? At what point, this is kind of an interrelated concept. I don't refer to myself as an aggregate of cells. I am an integrated and indivisible whole, and we'll stick with that definition for now. So I'm an individual. But why do people live as groups? Why do ants live as groups? What is so special about social living as a thing? When does this happen, and what triggers it, and why? So a group is technically in biology defined as a group of individuals from the same species. That's what conspecific means. Con meaning of the same, specific meaning species, who affect each other's fitness. So you and your classmates, you're a group. You affect each other's fitness. If one of you brings a virus to class, all of you get the virus and you get sick. You affect each other's fitness. Some individuals spend only part of their life as a group, and others spend their whole lives as part of a social group. And I've got a couple examples of this for you. So orangutans. Female orangutans actually live together as a social group for pretty much their entire lifespan, but male orangutans do not. They actually live basically off in the woods by themselves singly, and they only interact with the females for a short period of time. In fact, that period of time is basically mating, and it's about a 15-minute window. It is literally wham, bam, thank you, sir, now get out. And if the males don't leave when they're supposed to, the females, in fact, the whole cluster, the whole group of females, will turn on the males, they'll chase them down, they may even kill him and shred him. So he's got a really good incentive to get out of there. And that's why they tend to live singly off by themselves. Some of you are going, that's kind of harsh. Yeah, nature can be cruel. Other organisms like ants and honeybees are very social insects, and they spend their whole lives as part of a group. So it's all an integrated, very societal thing. And there's all kinds of different types of organisms with in-between schemas. So what we kind of have to tease apart is what are the benefits and the costs to group living? Because if we're looking at this from an evolutionary perspective, it has to have more benefits, hopefully, than costs, or natural selection should not favor it. So we're not going to look at all of these, but these are just some of the things that we can look at. Foraging and hunting behaviors, protection from predators. These are good upsides to being in a group. Child rearing is helpful, and we actually see that, although we don't go into it in this chapter, we actually see that among lions and other big cat species. Unfortunately, there are also some drawbacks, like the bigger your group, the more easily diseases can come through. Natural disasters can wipe out groups if they're clustered in one area. If you have to share your food, does that mean everybody gets less food? So these are some of the issues, and we're going to look at a few of those. And we start with spiders. Now, I know many of you do not like spiders, and that's okay. You do not have to love spiders, and this is not going to help. What we're looking at is a giant spider web, but we're also looking at a phylogenetic tree. So just focus on the tree. There's not even a picture of a spider here. It's not that bad. Okay, so all of these ones that are highlighted and that are represented with blue lines and a red dot, those are the ones that actually show group living. There are actually social species of spiders. So looking at this phylogenetic tree, question number 1, did the social living arise once and come from common ancestry, or did it arise multiple times? And I want you to answer that question for me on the next slide. Okay, hopefully you were able to reason your way through that. So what that tells us is that since this trait arose multiple times, the social living among spiders, is that natural selection favors this. Even if we don't understand why fully, we can see that there is obviously some sort of advantage, or it probably wouldn't keep popping up. So group behaviors are something that we're going to actually take a look at next. And in particular, we get to move away from the scary things, no more spiders, and we get to look at these little fish, bluegill sunfish. And we are trying to assess the benefits to this group existence. So to be beneficial, from a survival perspective, at least for this point, groups have to be able to get more prey, more food, than individuals could if they were hunting by themselves. Hopefully that part makes sense. So these fish actually eat aquatic insects. They're kind of hard to catch. And when the fish hunted together as a group, they caught more bugs. And the bigger the group, the more bugs they caught. Up to a point. It did eventually taper off. You run out of bugs. I mean, you can only flush out all the bugs. But what's interesting about this in particular is that this is a passive benefit. The fish in these groups didn't change their individual hunting behaviors, and that's what makes this passive. The fish hunted as though they were by themselves. So to put this in perspective, if you go tromping through the woods, if you go hiking by yourself, you're going to scare some critters in the bushes. But if a whole class of 30 of us goes hiking along the same path, we're going to scare off way more critters, and it's not because we're trying necessarily. It's just because so many more of us are that much more disruptive to the environment, and we scare off more prey. And that's exactly what's happening here. On the other hand, chimpanzees have a different system. They actually coordinate their hunting behaviors. So here's what happens with that. They have these chimpanzees that are drivers, and their job is to chase behind the prey in the trees and basically try to keep them moving in one direction ahead of them. That's their whole job, is just to chase. Those are called the drivers. So what's the prey going to do? Well, I don't know about you guys, but if there's something running behind me, I'm going to run ahead and maybe off to the side to try to escape. So some other members of the chimpanzee group actually climb up the trees and block them to prevent lateral escape. So now your poor little prey item, whether it's a squirrel or another monkey, which is unfortunately common, it can run in basically this direction forward, but it can't escape to the sides or behind. So since this is up in the trees, we still have to think in 3 dimensions. It could escape up only to a limited extent, and gravity says what goes up must come down, so eventually it's going to have to come down. Or it could go down and try to escape that direction. So there are chasers, which I know I don't like the way they named them, but that's not my job. They actually climb under the prey to keep it from escaping downward, and they'll occasionally try to snatch it and grab it as it's running. I'm sure the blockers do too. Okay. So now we've got this corridor. Everything's blocked along the sides, along the bottom. You've got something coming up after, behind you. You can really only run in one direction. And then at the very end, you have the ambusher that pops up and closes the trap. This is way more sophisticated behavior, and this one is a prime example of an active benefit to group living, because the individuals actually coordinate with each other, they change their behaviors, they hunt differently as a group, than they would if they were acting by themselves. Our next example are in honeybees, and this is an economy of scale in kind of a different sense than what we've looked at. This one has to do more with acquiring and transmitting information. So this is how bees actually hunt and forage for their food. Since they collect pollen, a bee will fly out from its nest, it will find a little patch of flowers, yay, all pretty. And once it has found the flowers, its job is to return to the nest and tell the other bees where the food is. So how does the bee communicate that? It actually uses a system of dance, and this is called the waggle dance. Not wiggle, waggle with an A. Spelling matters. Okay, so what the bee actually does is it will walk along this line in the beehive once it gets back to its fellows, and it will kind of wobble, it'll tilt back and forth as it does this, and it will kind of weave back and forth as it tilts, and it goes a certain distance, and then it will loop back around and it will start this waggle dance all over again. What this actually does is it communicates to the other bees where the food is, how far away the food is, and where the sun was when they found the food. So it's communicating, hey, that patch of flowers is 40 degrees that way as of noon. And the other bees can fly out using that information and find that exact same patch of flowers. This is pretty sophisticated nonverbal communication. What we found is that basically the farther away the food is, the longer the dance actually is. The longer this straight run actual part of it, where they're actually following along that particular tangent line. So if you need to communicate this patch of flowers was a really long trip, you're going to have to dance a little bit longer, as though you weren't already exhausted from flying. But this communicates to the other bees, it's over that way, here's where the sun was, and you're going to have to fly for quite a while before you get there, but the other bees can pick up on that information. And in the homework for this particular chapter, I have a podcast that tells you a little bit more about some other cool bee behaviors that also use the waggle dance. So I hope you enjoy listening to it. It's a really fun story, and the song kind of got stuck in my head. It will in yours too. I'm sorry, but it's interesting, even if you don't like bees all that much. They are fascinating little creatures to study. So we've kind of looked at hunting behaviors. We've looked at communicating where foraging sites are. This has to do with economies of scale in terms of being able to acquire and store and transmit information to be able to catch more prey. So economies of scale in those senses. And now we actually get to look at another one, and that's defensive capabilities. The many eyes hypothesis is really kind of based on the idea that if one group member detects a threat and runs away, then the other members of that group can follow and everybody wins. And this is true even if the bird or the white-tailed deer or the whatever wasn't responding any differently than it would alone. So this could also be passive. So if you know anything about white-tailed deer, when they get spooked, they run away, and they run pretty easily. Their tails flip up, and the undersides of their tails are actually white. This actually acts as a signal flare to other deer in the vicinity, and it basically says, I don't know what just happened, but something scared the crap out of me. I'm going to run away. Y'all should too. And then it's up to the other deer whether or not to follow. So this deer is going to run away no matter what. Whether its fellows choose to join it is up to them, and whether or not they actually catch the signal. Most of the time they choose to run. So this makes one kind of semi-reasonable assumption, and that's that the predator is only going to be able to catch one deer and not like the whole herd all at once. So this assumes your predator takes one prey item independently of the group size. This actually works best for intermediate sized groups. If your group gets too big, then it turns into a trample, Walmart at Christmas. If you've got smaller groups, there's still some competition, but it tends to work well because you get better, more efficient signaling among everybody. So a real world example of this was exploding fish. When I first read that title in the textbook, I really, really wanted to understand what was going on because I was kind of excited. Like, what do you mean? The fish actually like go and pop and like their guts go everywhere? That can't be evolutionarily beneficial. That's not what they meant by exploding fish. They got my hopes up. No, I'm not really a twisted individual. That's just the first thing that popped into my head. So what the exploding thing actually refers to is how they actually swim away from a predator. So it's a behavioral change that occurs with group living, particularly in fish that school together. And these are called flash explosions. So all of the individuals in this group, schooling around, oh, look, predator comes by, poof, they all swim off, each one in a separate direction. And this assumes, again, that the predator can really only chase one at a time. It's only got one mouth. So this actually helps to limit how many it's going to catch. Interestingly, this also seems to visually overstimulate the predator to some extent and cause some confusion. So they're going off in all different directions. The predator's just like, whoa, wait, what the bleep? Okay, and it misses everything. Now you may wonder about whether or not that's actually accurate, but one of the things that I want you to do just as a supplemental side project is pull up YouTube and watch a herd of zebras running. And I want you to see how difficult it is for you to visually, individually pick out a single zebra and follow it in that herd from somewhere in the middle of the herd. It's very hard for our brains to process. It's too much visual information. And that's actually exactly the point. That's part of what those stripes do, is visually confuse the predators, and it makes it harder for the lions or whatever who's hunting the zebras to actually zero in on one of them, target that one animal, and successfully attack that one animal. And it even works against humans. So that's pretty awesome. In fish, even just schooling behavior, swimming together as a group, tends to work similarly. So they actually sort of tracked this, and they did some computer models. They took some predator fish, these were sticklebacks, and prey, Daphnia, water fleas. And they actually looked at the group size. So if they had 5 water fleas, a certain number of them got, the targeting error, the distance from the strike to the nearest prey. Basically, how good was the predator at catching an individual? How much error was there? When the group size was small, like 5 water fleas, the predators were pretty good at catching them. When the group size got bigger, their error actually went up markedly. They became less able to target the fish in the group effectively, or the water fleas in this case. So this is partly because the visual systems, according to computer simulations, it was basically just too much input. It overloads and it causes confusion. And the animals can't just visually isolate and track one of the prey, just like the zebra stripes. So there are some definite benefits to group living, and that's kind of what we've been exploring in this particular case. You've got defense against predators, coordinated hunting behaviors, or even just passive benefits to hunting as a group. All of these things that natural selection should be going. Group living is absolutely the most awesome thing ever. Everything should live socially, but it doesn't. Not all animals live as social groups, and there are actually drawbacks too. So a lot of these apply when you live in a college life kind of situation. So you have to share your groceries, your laundry facilities, access to the bathrooms, you have no privacy. Okay, this is applicable for dorm life, apartment life, grown-up life with parents and kids. What you have is competition within the group for access to resources. If you've only got 1 bathroom and you've got 5 people, eventually somebody's going to have to wait. And that's just the reality of the situation. So that competition becomes an issue within the group. And there's another problem. Aside from competition, you also have increased risk of disease transmission. So this is something that's observed every year, every semester when school starts. People come back from vacation, they bring different pathogens with them, and a week or 2 after classes start, people get the back to school cold. It happens routinely. It's a very, very, very well documented phenomenon. It's also why you all were required to have meningitis vaccines when you came to college. And I was actually part of the meningitis, the reason that this actually occurred. When I went to college as an undergraduate at Radford University, we had a meningitis outbreak on our campus, and it was a particularly bad one. It affected faculty and students. They actually had dedicated meningitis response units. And it was very, very scary because there's not much we can do for meningitis once it happens to you. We can provide your body with supportive treatment, but especially if it's viral, we just kind of have to wait and hope for the best. That's the limit of our medical possibilities, is to make sure you get plenty of fluids and rest and hope you survive it. And a number of our faculty and students did not. We actually had several deaths on campus from this, and it was actually horrifying and terrifying. And if you don't know anything about meningitis testing, they actually have to do a spinal tap to confirm whether you have this or not. So if you developed a sudden severe headache, or if you had been exposed to somebody that came down with meningitis, you had to get tested, which means you had to go to the doctor, curl up in the fetal position, and let them shove a very large needle into your spine and draw fluid out, and it hurts. After that outbreak, the next year, they discussed whether we should make this mandatory for all schools, and that's why in the state of Virginia, the meningitis vaccine is actually required. It does not protect you against all forms of meningitis. In fact, I think it's only the viral meningitis. It does not protect you against bacterial or fungal. Part of that is because there's too many different versions of bacteria that can cause it. Fungal meningitis is actually pretty rare. A few years ago, we did have an outbreak in the US, and that was actually linked to contamination during drug manufacturing. And for some people, for management of chronic pain, they would actually get injections into their spine to manage chronic pain, and that drug was contaminated. That's where that fungal outbreak came from. So now that I've rattled on about that for a few minutes, how does this play out? Our kind of last example are these little cliff swallows, and these are little birds that tend to live together in social groups. And what's kind of interesting is that we can actually track their survival, and the smaller groups that only have maybe 10 birds or so, you'll notice their survival in the 10 to 50-ish range, kind of in this region, is somewhere between 50 to 80%. Crappy. The larger your colony gets, the better your survival gets. Once you get up to a colony size of 75 birds or so, pretty much all of them have a good chance of surviving. It's almost 100% survivability. And it stays that way until your group size hits about 1,000, and then it actually drops down again a little bit. Not a huge amount. So there's clearly a benefit to larger group size up to a point. And once you get past that point, we start seeing more and more of the drawbacks. So the questions really are, what's the difference? Survival obviously improves with bigger groups, but what about reproductive success? Here's another interesting part of the question. So what we're looking at in this particular graph is, again, colony size, how many birds are in your group, and the number of unhatched eggs, or basically the percentages of nests that have more than one unhatched egg. The bigger the group, the more eggs actually hatch, the fewer unhatched eggs you have. So survival, that benefited from group size. Reproductive success, that one benefited from increasing group size. So selection faces a bit of a trade-off, especially if you get into these large groups. You can have more eggs that hatch, but the survival at this point was right at that point where it started to drop back down a little bit. And here's why. They also tracked the number of insects, the number of parasites per nest. And these are actually swallow bugs. They're kind of related to bed bugs. These are blood-sucking little parasites, and the more birds you have in your colony, the more parasites you have in your colony, especially as you get above about 1,000. And since these are blood-sucking insects, they can transmit blood-borne illnesses, which accounts for that slight drop in fitness and survival after that initial really good level in the 100 to 1,000 bird range. So in this chapter, what we've kind of traced out is how we got eukaryotic cells, a little bit about tissue differentiation and the origins of sexual reproduction, which was in there. Those tissue differentiations were necessary for sexual reproduction. Why things live as groups, what some of the benefits and drawbacks are, and this helps explain why some species live as groups and socially and others do not, why some organisms are single-celled and why some are multicellular, and why we have so much diversity in terms of that. It's because of how we address these kinds of trade-offs. In the next chapter, we actually get to look at something that's almost completely different, and chapter 13 is honestly one of my favorites. So stay with me. I'll see you soon.